Hi, everyone who's tuning into the webinar now. We are just going to wait a few more minutes to get all on board, and then the webinar will start very soon. Okay, I think we will start the webinar now. So welcome everyone to the first webinar of the ISHR European Section webinar series, The Beating Edge. And the topic of today's webinar is cardiac metabolism. We have an excellent lineup of speakers that we are very lucky to get to listen to, and the webinar is presented to you by the early career investigator representatives of the ISHR-ES, Dr. Jonathan Lambert from the University of Padua, and myself, Dr. Caroline Rypdal from the University of Oslo. And I would just like to mention for everyone that the webinar today is being recorded. However, it will not be published online before all the data from today's speakers are published. So that may take a while. So just before we begin, we would like to briefly mention some important information and upcoming events of the ISHR. And first and foremost, you should all make sure to become members of the ISHR-ES. If you do not already have an active membership, the membership will provide you with benefits such as reduced rates for the ISHR conferences and events, and also eligibility for awards and fellowships within the society. And of course, we would like to mention the upcoming ISHR Congress, which is in Nara in May. And there is one important deadline here that you should note, which is March 15, where we have the abstract submission ends and the deadline for application for the Early Career Investigator Travel Awards, which was extended. And we also have the early bird registration discount ending on March 15th. So you should all make sure to register before March 15th. And then we are really happy to announce that the next, ISHR-ES European meeting will be held in Birmingham in 2026. So there's currently a proposal for symposias for this event. So if you have an idea for a scientific session, you can fill out the online application form for Birmingham 2026. We are also or we would like to mention our two journal, which is a Journal of Molecular and Cellular Cardiology, JMCC, and the new the JMCC Plus journal. And this journal just got accepted for indexing in all databases and will receive its first impact factor in June. So this is really exciting.
So it's Jonathan here. So if any of that was news to you, we highly encourage you to follow us on the European section social media accounts. We have a new LinkedIn page, and we just launched a Blue Sky account, and we're still very active on Twitter and X. So please make sure to follow those accounts to stay up to date with European section activities and for future webinar session announcements. So looking forward, our next session will take place after the World Congress in Japan, and it's going to focus on the theme of cardiac fibrosis. And in future sessions, we're going to feature early career and mid-career investigators as speakers, and we welcome speaker suggestions. So if you're interested, please get in contact with us. So those are really all the latest updates, and let's really just dive into today's session. So I'm just going to give a quick overview of our format. First of all, thanks again for joining us. We have an exciting lineup for our first session of the beating edge of cardiac metabolism. First, we're going to hear an introductory talk from European Section President Christoph Mack. That will be 10 minutes in length on the clinical aspects of HFpEF and metabolic diseases. And we will then move on to our invited speakers and leading cardiac metabolism experts, Professor Lisa Heather and Professor Timothu Wai. Their talks will be 15 minutes in length with 5 minutes of question and answer time. We highly encourage everyone to submit questions on the Zoom chat box, Q&A box. And following the speakers' presentations, we will then relay those questions to the speakers for answers. So we're ready to get started. So Christoph, if you could go ahead and share your slides as I briefly introduce you. So our first speaker really needs no introduction because he's the European Section President, Christoph Mack. But Christoph earned his MD PhD from the University of Cologne before he completed a postdoc in Brian O'Rourke's lab at Johns Hopkins University in the US. Since 2017, Christoph has been the spokesperson for the Comprehensive Heart Failure Center at the University Clinic in Würzburg, Germany, where he also chairs the Department of Translational Science. His research focuses on the cellular defects in chronic heart failure, and he's particularly interested in investigating contractile, mitochondrial, and metabolic dysfunction. And his encyclopedic knowledge of the field will now take us through the beating edge of the clinical aspects of HFpEF and metabolic diseases. So thank you for joining us, Christoph, and the stage is yours.
Yeah, thank you, John, for the nice introduction, and thanks to all of you for organizing this, also to Caroline and Davo, of course. So I will just give you a brief overview on the metabolic aspects in heart failure, particularly with preserved ejection fraction. So as you know, the global burden of heart failure is pretty large, and especially in Germany, we have a high burden right now. And looking into Germany, we see, but also in other countries, an exponential increase in the prevalence of heart failure with age. So it's an age-dependent disease. So meanwhile, our median age is 76 years, and more than 50% of the patients are female, and they come to the hospital about once per year. But in 3/4 of the cases, this is a non-cardiovascular hospitalizations. Which tells you a lot about the comorbidity burden on those patients with heart failure. And this is another analysis from Würzburg a couple of years back already. In the early 2000s, more than 50% of patients with heart failure had 7 or more comorbidities, and the number of comorbidities would predict a poor prognosis in these patients. And this comorbidity burden has been increasing ever since the early 2000s up to more recent years. And this is an analysis from the United Kingdom that also illustrates this fact. So heart failure is generally been seen as a cardiovascular continuum where the risk factors such as diabetes and hypertension, but also others, can on the one hand induce coronary artery disease, which can lead to myocardial infarction and the loss of functional myocardium. And this typically leads to heart failure with a reduced ejection fraction or HFrEF. But in about 2/3 of these cases of HFrEF are having an ischemic etiology, whereas the other 30% roughly, 30 to 40% have other causes of cell death, such as myocarditis, chemotherapy, or other factors, but also hereditary causes can play an important role, and they can add to each other. But meanwhile, more than 50% of our patients with heart failure have a preserved ejection fraction. And here, typically, these patients also have similar comorbidities, but are generally elderly and more often obese. And here is a short overview on how patients with HFrEF and HFpEF differ. Here you see the different factors in relation to the left ventricular ejection fraction category. And you can see that patients with HFpEF are older, are more often female, have lower NT-proBNP levels, and have much less often ischemic etiology. The prognosis is comparably poor, but this is also related to the fact that patients with HFpEF are older. And if you have a closer look at the underlying causes of death, you can see that patients with HFrEF have more often cardiovascular death compared to HFpEF, and HFpEF patients have more often a non-cardiovascular death. This way or the other, patients with heart failure with any etiology have a very high comorbidity burden, and especially obesity, diabetes as metabolic diseases are very common, but also chronic kidney disease and anemia. So this tells us that heart failure is a systemic disease where the heart is in communication with other organs. And the question is, how do these organs communicate with each other? And we have at least three important pathways. One is the neuroendocrine activation, one is immunity and inflammation, and the third one is metabolism. And so far, heart failure treatment has much focused on antagonizing the neuroendocrine activation with compounds such as ACE inhibitors, beta blockers, aldosterone antagonists, and so on. However, most of these treatments are not efficient when it comes to an ejection fraction of 55% or above. And this is the blank spot where we are still trying to improve our treatments. And in the recent years, we have seen breakthroughs with the treatment of compounds that are targeting metabolism, such as SGLT2 inhibitors and GLP-1 receptor agonists. And I'd like to briefly touch upon these two compounds. First of all, the burden of obesity has been increasing since the 1960s and is still on the rise in all of the countries in the world. And this also leads to an increased burden of diabetes, which Lisa will focus a bit upon. Here you see the increase in numbers in the different continents of the world, and everywhere it's still on the rise. So heart failure and diabetes have a close interaction with each other. Namely, you have neuroendocrine activation in heart failure, and this can lead to sodium and water retention on the one hand, but on the other hand, also induce through sympathetic activation lipolysis, plus the hypernutrition that you have leads to obesity and insulin resistance, which forces the pancreas to produce more insulin, which it can do for a certain amount, but eventually it will decrease the insulin secretion, and this at the latest will cause hyperglycemia, which is a risk factor for especially macroangiopathic diseases, which will lead to myocardial infarction and rather a HFrEF phenotype, but also fibrosis, inflammation, and microvascular dysfunction are the consequence of this diabetes, and this can then contribute to the particularly characteristics of heart failure with a preserved ejection fraction. And many anti-diabetic drugs can reduce the blood sugar, but they do this by very many different mechanisms. And in this context, not all of them are successful, but we have some which are rather adverse, such as the glutazones and likely also insulin. But we have other compounds such as the GLP receptor agonists and especially SGLT2 inhibitors, which have shown great potential or actually evidence for protection in patients not only with diabetes but also heart failure. So on the mode of action of SGLT2 inhibitors, it acts through secreting glucose from the body, and this can lead to a diuresis. And with it comes a little bit of decongestion and a little bit of blood pressure lowering, which are in principle positive effects for the overloaded heart. It's also leading to nephroprotection, particularly through lowering the glomerular filtration pressure. But it also has a substantial metabolic effects, and by the elimination of glucose, it can lead to more lipolysis, where the fatty acids go to the liver and are formed to ketones. And many people consider ketones to be an additional and rather beneficial substrate for the heart. But also on a cellular level, it's been shown that the SGLT2 inhibitors can increase SIRT1 and PGC1 alpha signaling, which are important regulators of biogenesis to make new mitochondria, but also autophagy, which is important for the cell repair. But finally, since it's also been observed that patients with diabetes benefit just as much as patients without diabetes, it's still been discussed whether off-target effects independent of SGLT2 can have beneficial effects. And in this context, inhibition of various sodium transport systems, such as the late sodium current or NHE, could be positively affected by SGLT2 inhibitors. The clinical data in HFpEF are quite clear. We have 20% risk reduction for hospitalization for heart failure and cardiovascular mortality, which is mostly driven by a reduction in hospitalization more than of mortality. And the benefit goes across the spectrum of ejection fraction, which means that irrespective of whether you have HFrEF, MREF, or PEF, you can treat these patients with SGLT2 inhibitors, and therefore they receive class 1 recommendations for PEF, MREF, and they already had it before for the reduced ejection fraction. And finally, obesity is on the rise, and the increase in body mass index and obesity increases the risk of developing heart failure with a preserved rather than a reduced ejection fraction. But on the other hand, once you have, or furthermore, once you have heart failure, again, also here, obesity will increase the risk for a worsening heart failure event, but not so much for mortality. The good news is that with prevention, such as exercise, you can reduce the risk of HFpEF, not so much of HFrEF. And meanwhile, we also have compounds that target obesity very specifically, such as the GLP-1 receptor agonists. And GLP-1 is an endogenous hormone, which is released by the enterocytes and will induce by paracrine, neuronal, but also endocrine pathways, various effects in the whole body. And I'd just like to point out that it's been developed for the development of diabetes by increasing insulin secretion, but it's also having substantial effects on the brain and the stomach, which lead more or less to the fact that there is an earlier saturation after food ingestion, so that people just eat less. And the effect is that patients with HFpEF, which are treated with GLP-1 agonist, lose body weight about 10% and have an improved quality of life regarding the KCCQ index, which is irrespective of having diabetes or not. And there have also been meta-analysis looking at the harder endpoints with semaglutide. It looks like it's also reducing the combined endpoint of cardiovascular death and hospitalization for heart failure, which is again driven mostly by heart failure events and not so much by a reduction in mortality. And finally, there is a new compound called tirzepatide, which is a combination of a GLP-1 receptor agonist and gastrointestinal peptide GIP receptor agonist. I will not go to the details, but it has a synergistic effect on obesity. And what the clinical effect also here is a reduction in the combined endpoint of cardiovascular death and hospitalization for heart failure in patients with HFpEF and obesity. And also here, the endpoint is mostly driven by reduced hospitalization and less of mortality, but also these patients experience an improvement in quality of life. So in conclusion, after decades of research on neuroendocrine blockade, we are now entering an era of metabolic treatments where SGLT2 inhibitors and GLP-1 receptor-based treatments are the spearhead of development, and they have shown particularly benefit in patients with heart failure with a preserved ejection fraction. So thank you for your attention, and now we're looking forward to the exciting talks by Lisa and Tim.
Thank you very much, Christoph, for that really interesting introduction on cardiac metabolism and for setting the stage for consequences for the heart. We will not have questions for you, unfortunately, for the sake of time here, but for our next speakers, we will allow questions in the Q&A box. So we encourage everyone to ask questions for the speakers. And now we move on to our first speaker, who is Lisa Heather. She's a professor or associate professor and British Heart Foundation Fellow at the University of Oxford. She received her PhD in cardiac metabolism in 2007 from the University of Oxford, where she continued her research and currently runs a research group studying the effects of abnormal metabolism in the type 2 diabetic heart. Her work has demonstrated that accumulation of specific metabolites within the myocardium results in dysregulation of numerous signaling pathways that drives cardiac dysfunction in diabetes. And Lisa has been the recipient of several awards for her groundbreaking work, including the ISHR Outstanding Investigator Award at the European meeting in Toulouse last year. And now we really look forward to hearing your talk, Lisa, entitled Hijacking Metabolite Signaling in Type 2 Diabetes. Please.
Thank you so much for the really kind introduction. Can I just check you can see my screen and you can hear me okay?
Yes, perfectly.
Yeah, perfect. Thank you very much. Okay, right. So the healthy heart sits in what we can describe as a metabolically privileged position, what we call the Goldilocks zone, because it has evolved to use the right range of substrates to metabolize them at the correct rate, to make the perfect amount of ATP to power cardiac function. And although we describe the heart as being a metabolic omnivore because it can use a range of different fuels, we know that there is an inherent metabolic preference within the heart. And it's been estimated that 60% of the energy needed to power contraction comes from the metabolism of fatty acids, which are taken up from the circulation. The remaining 40% of energy come predominantly from the metabolism of glucose, but also from the metabolism of ketone bodies, amino acids, and lactate. And it's this balance between these substrates that power the heart to work under healthy conditions. So what's been known for decades now is that if we force the heart out of this Goldilocks zone, out of this optimal metabolic preference, it results in impairments in many processes, including contractile function. So if we force the heart to using more sugar, so if we force it to the left in this diagram, we end up with a heart such as what we see in heart failure with reduced ejection fraction, where we see compromised function. And if we force the heart in the other direction, so if we force it to use too much fat, we end up with compromised cardiac function, as we see in type 2 diabetes. So the heart has a happy zone, and if we force it in any direction out of this from a metabolic perspective, we end up with disease. Now, my research group is really interested in what happens in type 2 diabetes, and we study what happens very early on in the disease progression. So before establishment of HFpEF or HFrEF, we're really interested in saying what happens early in disease process. And the reason for this is we're trying to unpick what the role of metabolism is in this impaired cardiac function. And type 2 diabetes is your quintessential metabolic disease. We start with abnormal metabolism, and in time we end up with abnormal function. So this model allows us to unpick the role of metabolism in disease. So what happens to metabolism in the type 2 diabetic heart? The answer is early in the disease progression, we see a clear metabolic shift in the preference for fuels within the heart. So the metabolism of glucose within the cardiomyocyte is downregulated in type 2 diabetes. And this is a bit counterintuitive because we know that the blood is hyperglycemic. We have an increase in blood glucose levels. But at the level of the cardiomyocyte, the glucose metabolism pathway, so glycolytic flux and pyruvate oxidation as assessed, as a surrogate marker for glucation are both downregulated. So as the heart is using less glucose, it's shifting towards using more fat. So more fat is being taken up from the circulation and is being oxidized to generate ATP, and more fat is accumulating as myocardial triglycerides within the cytosol. So early in the disease progression, the heart is rebalancing its metabolism to shift away from glucose use towards using more fats. And this shift in metabolism has consequences for the function of the mitochondria and for energy generation. So we see decreases in mitochondrial respiration, small decreases to start with, but we see impairment in mitochondrial function. And this results in an energetic defect in the heart. So using phosphorus spectroscopy on isolated perfused hearts, we can quantify the amount of energy in the intact contracting organ, and we see decreases in AT and phosphocreatine before we see any kind of changes in systolic function. So it's clear that there's an energetic defect occurring early. But it seems also that the diabetic heart is sitting on a little bit of a knife edge. It can cope at baseline, but if we push it, it really can't respond. So this was measuring phosphorus, using phosphorus spectroscopy to measure the amount of energy in the heart in response to a myocardial ischemic challenge. And what you can see is the energy defect at time 0 when these hearts are normally perfused is not that different between the controls and the diabetics. But as we challenge them with ischemia, you can see that the energy levels in the diabetic heart drop off much more rapidly than in the controls. They simply can't respond metabolically to maintain their energy production in response to this stress. So at this stage of the disease progression where we're seeing already this metabolic shift, this energetic dysfunction, if we look at what's going on with contractile function, what we find is that systolic function is perfectly well maintained. These hearts have got really good systolic function. But we already see evidence of diastolic dysfunction emerging. We find that the diabetics have evidence of impaired diastolic dysfunction. We can see this via echocardiography. And we also find with regard to cardiac function that they're just about coping at baseline, but if we challenge them either with an ischemic challenge or with a hypoxic challenge, what you can see is these diabetic hearts simply cannot recover to the same extent. So they have lost this inherent flexibility to be able to respond to challenges. So at this stage, we can say, okay, we have abnormal metabolism in type 2 diabetes. We are seeing evidence of abnormal function, either diastolic dysfunction or this impaired response to a challenge. But at this stage, we can't make this connection. We cannot unequivocally say that it's the abnormal metabolism that's driving this abnormal function. So one thing my group's been doing over the last probably 10, 15 years is we've become really interested in the concept of metabolic therapy, to ask the question, if we correct the metabolism, do we end up correcting the function of these hearts? So I just wanted to highlight 2 studies that we did, which I think really make this link between metabolism and function in diabetes. So the first study we did is we decided to try and increase glucose metabolism. So we took diabetic animals and we treated them with this compound called DCA, which opens up the pyruvate dehydrogenase reaction, which is the link reaction between glycolysis and glucose oxidation, to see if we could upregulate glucose use within these hearts. And to assess this, we used an in vivo magnetic resonance spectroscopy technique called carbon-13 hyperpolarization, which allows us to measure glucose oxidation in vivo in real time in these rats. What you can see in the diabetics is that, as we predicted, they have decreased PDH flux, so they have less glucose oxidation, and this goes hand in hand with diastolic dysfunction. But when we use this DCA to open up glucose oxidation, we can restore PDH flux back to what we see in the control animals, and we basically return the diastolic function back to what we see in the control heart. So this is really saying correcting glucose metabolism can correct the function of the heart. But equally, we can do the opposite thing. We can try and block the fat metabolism. And by blocking the fat metabolism, we used a compound called SSO, which prevents fats getting across the sarcolemma and into the cardiomyocyte. And what we showed is that if we take diabetic animals and we block fats coming into the heart, we can reduce the fatty acid oxidation rates back to what we see in the control animals. And when we do this, we can restore this response to stress, so that we can restore the systolic function that we know is impaired in response to stress in the diabetics, and we can bring it back to what we see in control levels. So what this is really telling us is that the changes in metabolism that we're seeing in diabetes are directly responsible for the abnormal function we're seeing at this stage in the disease progression. And therefore, correcting metabolism does seem a viable target for correcting function in diabetes. But as with all research, you answer one question and it raises another question. And that was, well, how is metabolism doing these things? Like, you know, how is metabolism having these negative effects on the function of the heart? What is actually going on at the level of the cardiomyocyte to mechanistically link these two? And this is something we've been thinking about probably for the last 6-7 years now. And I think if we take a traditional view of metabolism and what metabolism can do, and if we focus on the lipids, which we know are accumulating in the myocardium, we know there's more fats coming in, they're accumulating at the level of the cytosol as triglycerides, they're accumulating within the mitochondria. What are these guys actually doing? And our traditional view of lipids is that they are sources of energy, they are structural molecules, they are storage molecules. But what my group has really been working on is trying to think of alternative roles for lipids. And one of the things that we've identified over the last few years is that lipids can be potent signaling molecules. And that in diabetes, when we get more of these lipids accumulating, we start to hijack these signaling processes within the diabetic heart. And this is having deleterious effects on the cardiomyocyte. So we've shown that when we accumulate lipids in diabetes, we start inappropriately regulating transcription factors. And we've shown this for the transcription factor, the hypoxia-inducible factor, which we know is critical for survival post-ischemia. We've shown that lipids can be substrates for post-translational modifications, and therefore we can have impacts on various post-translational modifications through the cell. And we've also shown that lipids can have an acute effect, competing with the regulation of enzyme activity, and this can have acute effects, for example, in response to ischemia. So it seems that lipids and lipid accumulation that we're seeing in diabetes isn't just a traditional metabolic effect, it's doing other things as well. So what I wanted to do for the second half of my talk is talk to you about totally unpublished data where we've been looking at a new lipid-driven post-translational modification. And as I said, this is unpublished data. It's the first time I presented this work. So we became interested in a protein called fatty acid translocase, or CD36. And this is a protein that is responsible for bringing fat across the sarcolemma into the cardiomyocyte. So a way of viewing it is this is like our metabolic gatekeeper. It brings the fat in, it allows it to accumulate, and then it allows it to either go into storage or oxidation. So really, it's the primary step in all of fat metabolism within the cardiomyocyte. And what's interesting about this protein is it exists in two main locations within the cell. So it can be intracellular within vesicles, and under this condition, it is inactive, or it can be trafficked to the membrane where it then becomes active and brings fatty acids into the cardiomyocyte. So depending on its location in the cell will determine whether fats are coming in or if that process is suppressed. And something that has been known for a number of years now, and we confirmed in our animal model, is that in type 2 diabetes, we see a relocation of this protein to the sarcolemma. So in diabetes, you have more CD36 at the membrane. And the way I view this is it's a bit like opening a tap at the membrane and just allowing lots of fats to flood into the heart. And we became interested in trying to understand why this is. Why are we seeing more CD36 at the membrane? And is this underpinning a vast majority of the metabolic changes we see in diabetes? So we started looking at a post-translational modification called protein S acylation. And this is the addition of a lipid intermediate, so a long-chain fatty acyl-CoA, to a cysteine residue of a protein. And when this long-chain fatty acyl-CoA gets added on, this is associated with moving proteins to and from membranes. So addition of this long-chain acyl-CoA moves proteins and allows them to become embedded in the membrane. And this is an enzymic process. It's regulated by palmitoyl acyl transferases, which are called ZDHHCs. And these move the protein to the membrane, and it can be removed by depalmitoylases, which are called APTs. And our thought was, okay, in diabetes, we've got more of this protein at the membrane, and we've got more of the substrate for this post-translational modification in diabetes, this long-chain acyl-CoA. Could these things all be linked, leading to what we see in diabetes of this lipid overload? So we first looked in diabetic rats and then followed this up in diabetic pigs and then subsequently in human iPS cardiomyocytes that were insulin resistant. And we showed that in all of these diabetic models, there is an increase in this post-translational modification happening on CD36. So there is an increase in CD36S acylation. And we then started to think about, okay, if we've got more of this post-translational modification, we've got more of it in the membrane, what is driving this? What is mechanistically responsible? And as I said, there are enzymes involved in this process. So there's 2 enzymes that add this acylation group. These are the ZDHHCs. And then there's 1 enzyme that can take it back off, which is the APTs. And we showed that in diabetes, there is an upregulation of one of these key enzymes that adds the acel group on, the ZDHHC4. And this is localized at the level of the Golgi and is responsible for adding the acel group onto the protein in the first place to get it moving towards the membrane. So we thought, well, maybe this increase in ZDHHC may underpin the changes in CD36 localization at the membrane. So we looked at the promoter region of ZDHHC, and this was work carried out with a collaborator here in Oxford, Professor Sarah Duval. And she noticed there are a number of transcription factor motifs within the promoter of this protein, this gene, the ZDHHC4. But the one that jumped out to us was there was a motif of FOXO1. FOXO1 is a transcription factor that we know is upregulated in diabetes, and we know it's involved in reprogramming metabolism. So we started to question in cellular models, maybe it's activation of FOXO1 that's driving this increase in ZDHHC4. So in cellular models, working closely with a collaborator in Alberta, Professor John Usher, we were able to show that FOXO1 does bind to the promoter region of this ZDHHC4. And if we knock down FOXO1, we decrease the expression of ZDHHC4. And if we decrease ZDHHC4, we decrease this acylation of CD36. So at least on the cellular level, we seem to have a working model where we think FOXO1 is regulating the transcription of ZDHHC4, and this is driving this post-translational modification on CD36. But obviously, we wanted to confirm this in animal models. So we were fortunate enough that our collaborator in Alberta had a FOXO1 cardiac-specific knockout mouse. And what he was able to show for us is that if we take a normal MHC cree mouse and we make it diabetic, we see an upregulation of this ZDHHC4 acylating enzyme, and this results in an increase in CD36 acylation. But if we do the exact same experiment in a FOXO1 knockout mouse, we prevent this response. Similarly, we showed this via a pharmacological response that if we inhibit FOXO1 in diabetic animals, we can reverse these changes. So we can take back down DHHC4 and CD36 acylation. So this left us with a working model in diabetes where we are saying that activation of this transcription factor FOXO1 increases the expression of ZDHHC4, our acylating enzyme, which results in an increase in S-acylation of our target CD36. Well, this was fine. We then had a mechanism to explain what was going on, but we wanted to then link this to the significance in diabetes. Is this an important process in diabetes? Can we modulate it to improve the diabetic heart? And working with collaborators at the University of Chicago, they had developed a DHHC inhibitor called CMA. So we said, can we give this inhibitor of this process to diabetic animals and see if we can reverse the metabolic changes we're seeing in diabetes? Because this will tell us whether this CD36 S-acylation is actually important to the pathology. So what we showed was infusing this DHHC inhibitor into the diabetic setting, we were able to decrease the S-acylation of CD36. And this resulted in moving CD36 away from the sarcolemma and into the intracellular vesicles where it's inactive. And the consequent effect of this process was that we were able to decrease the fat oxidation in these hearts, decrease the myocardial triglyceride stores. And the knock-on effect on cardiac function was that we see an improvement in the rate pressure product in these hearts from these diabetic animals. So although these animals are still diabetic, we have corrected the metabolic processes that we think underpin the contractile dysfunction. So to finish off, I always like kind of doing one side of the story and the other. So we've said we can make a diabetic heart better by suppressing this acylation. Well, what about if we do the opposite? What if we take a healthy heart and we ramp up acylation? Does that make the heart unhappy? So we used a compound which inhibits the deacylation. So it means more proteins in the cell will be S-acylated. And what we showed is that taking a healthy heart and infusing this compound was able to increase S-acylation as we hoped it would, increasing CD36 S-acylation. And this resulted in more CD36 being localized at the sarcolemma. This resulted in more fat coming into the heart and being oxidized, and a trend towards an increase being stored within the myocardium. And critically, when we did this, we start to see impairment in cardiac function with this borderline decrease in rate pressure product. So hopefully what I've managed to convince you today is that in diabetes, when we see this overload of lipids, we do impact all the traditional kind of views of metabolism. So we do impact energy, storage, and structural roles. But fats are clearly signaling molecules regulating other processes. And by understanding how they impact these signaling processes, we might start to understand why in diabetes we see these changes in cardiac function and allow us to identify new targets for therapy. So this is really the people who did all the work. I just get the privilege of presenting the data, and I'd like to thank my collaborators as well. I always seem to take pictures of my group at Christmas. My photos are always got a Christmas tree in, but maybe it's always Christmas here in Oxford, so that can be my excuse. But thank you very much for your time.
Thank you very much, Lisa, for sharing your new and interesting data. That was really interesting to me. I'm not involved in cardiac metabolism research at all, and now I really want to be, so thank you for that.
One comfort.
I think just while you or the participants can put their questions in the Q&A box, I have a question for you, first of all, because this diabetic cardiomyopathy or diabetic heart, that's, I guess, is a main risk factor for developing HFpEF. And would you say that this pathway may be a target for new medication for HFpEF patients?
Yeah, I think we're looking earlier on in the disease progression, and I think a lot of what we're seeing is kind of almost preloading the heart as it progresses into HFpEF. I think one of the challenges with metabolism is it's so interlinked, is working out what our target should be, which pathway or which protein should we specifically go after. And I think we need a metabolic target that is in some ways unique to the heart and not, all these processes are shared between different organs. So I think there's some real interest, and I think particularly these DHHCs have cardiac specificity. The isoforms are more prevalent within the heart than in other tissues. So I think that can give us an angle with regard to what we should be targeting metabolically, but we'll see.
Thank you.
Hi, Lisa. Thanks for the great talks. We actually have a couple of questions in the chat. The first one comes from Danielle Hernandez. It says, very interesting. Do you know or hypothesize that CD36 would be decreased in other models of heart failure, ischemic chemotherapy, where fatty acids are decreased?
So we started off studying HFrEF, and in HFrEF, CD36 is downregulated with all the proteins involved in fat metabolism. So it is the opposite story to what you would see probably in HFpEF and certainly in diabetic cardiomyopathy. So yeah, it does seem to be a key step, this kind of regulation of CD36. We've also looked in aortic stenosis, and it goes down in there. So I think kind of when it's reduced ejection fraction, I think we're seeing decreases in CD36 along with other proteins involved in fat metabolism.
Okay, thank you. So the next question comes from Kate Harum. And this is interesting. So she asks, does the diastolic dysfunction, could it be due to metabolic changes from the cardiac fibroblasts?
Great question. And yeah, we look at the heart as a whole. We're not looking at individual organs. The only thing I can say is we've got a paper under review at the moment where we can see diastolic dysfunction in engineered heart tissue that we've generated from iPS cardiomyocytes. And that is obviously a cardiomyocyte enriched population where we think there's less fibroblasts. So watch this space and we might be able to have a better idea. But certainly in the EHTs, we still see the diastolic dysfunction.
All right, so it still needs some research. And there's one more from Thomas, and he asks, how specific is ZDH towards CD36 acylation? As recently, ZDH11 was shown to acylate ATGL. Are there other substrates of ZDH? And I guess I'll extend that. Could it possibly be affecting contractile machinery? And this is why you see the diastolic dysfunction.
Yeah, Yeah, great question, Thomas. Yes, so it seems that 4 and 5 seem to show specificity for CD36, but we don't know about other targets. I think the field is, making rapid advances in the moment of identifying all these different targets, but I don't believe for a second that this is a CD36 exclusive effect. I think it's probably doing things to other targets that we're not on top of yet as well.
Okay, and we have one more before we let you off the hook. So from Christian, very nice talk. In the diabetic model, is FOXO upregulated to begin with, or is it activated in response to elevated lipid uptake, which in turn leads to the ZDH upregulation, which moves CD36 to the membrane?
Yeah, I mean, FOXO1 is being activated by something, isn't it? I mean, that's what kicks off the whole process. I'm not as clued up on what the drivers for FOXO1 activation are, but I think potentially targeting the activation of FOXO1 would be a way of rebalancing multiple aspects of metabolism, because FOXO1 regulates multiple pathways, not just obviously the DHHC4.
Okay, well, thank you very much, Lisa. That was a terrific talk, and thank you everyone for the very interesting questions. We'll move on right to our next speaker. So Tim, if you could go ahead and start sharing your slides as I introduce you. So next we have Professor Timothu Wai. He earned his PhD from McGill University in Canada before completing a postdoc in Germany in the lab of Thomas Langer at the University of Cologne and the Max Planck Institute of Biology and Aging. Since 2010, Tim has led the mitochondrial biology unit at the Pastuer Institute in Paris. And he truly is a mitochondrial biology expert and has made groundbreaking discoveries in the field of mitochondrial dynamics, particularly identifying novel suppressors of mitochondrial fragmentation. His research extends across multiple disease settings, and he looks at mitochondrial dysfunction in liver, neuronal, and cardiovascular biology. He even studies how microbes and pathogens interact with mitochondria to evade exploit cellular mechanisms in infectious disease. But today, Tim will be talking about the cutting edge of mitochondrial proteostasis in cardiac health. So thank you very much for being here today, Tim, and the stage is yours. You're still on mute, Tim.
Let's try this again. Can you hear me now?
There we go.
All right, we're
I can
cooking.
hear you now.
We're cooking with gas. So thanks, Jonathan. What I'm going to do today is tell you a totally unpublished story that we honestly don't fully understand, but that's part of the fun. It starts with obviously mitochondria, which is what my lab is focused upon. I'd like to highlight that mitochondria do lots of metabolism, energy metabolism, biosynthesis, but they also have important signaling roles. And we try to figure out which ones are important, when and where and how. We're convinced mitochondria are important, but how they're playing roles in disease and normal physiology is for us complicated to understand at times. When we think of energy production, and Lisa touched upon this, we think often about the oxidative phosphorylation system, in which redox equivalents are driving electrons through complexes 1 all the way to 4, where oxygen is consumed. And in this process, protons are pumped in the IMS. And these protons are harnessed to generate ATP from ADP and inorganic phosphate through the ATP synthase. And this is certainly in the heart and important to be a coupled process, so that the proton pumping and that the energy generation or the ATP synthesis is coupled. And so any kink in this chain could be really catastrophic in the context of energy production. So what I want to tell you about is work that's been done in the lab studying mitochondrial dysfunction in an animal model and how this impacts the signaling function. We're trying to figure out which is important in the context of the disease and notably in the heart. But the story actually doesn't start in the heart. The story starts in a type of neurological condition that has ALS and frontotemporal dementia. So it's a motor neuron disease combined with dementia that's adult onset and happens in patients that carry a heterozygous mutation in a mitochondrial protein that I'll tell you about called CHCHD10. Now this work started with Véronique in Nice, who runs a clinical genetics reference center and identified the first French families that had these heterozygous mutations. And what these patients presented with were, beyond the ALS and the FTD, were in the cells that they could get from the patients, were cytochrome oxidase deficiency, so an inability to consume oxygen properly in the muscle fibers, defect in ATP synthase, loss of mitochondrial DNA integrity, and defects in cristae, which are the intermembrane invaginations in which most of the OXPHOS machinery is located. There's a bunch of mutations in D10, but the one that we're going to talk about is S59L, and this happens to be in this coiled central alpha helix region of this tiny little protein, and it's heterozygous. Most mitochondrial mutations are recessive in nature, but this is a gain-of-function mutation, and there's been work that's been done in her lab for the last 10 years studying the impact of this. What's fascinating is that D10 is a protein that is made outside mitochondria, like most mitochondrial proteins. It finds its way into mitochondria through an import channel on the surface of mitochondria, and there's a disulfide relay involving MIA40 nerve 1 that's involved in taking this linear protein and folding it. This is really, really important. I won't bore you with all the details, but basically there's some oxygen that's consumed, electrons are transferred. But if you don't have this system, this protein doesn't get folded properly. Now, it's not just D10 that goes through this route. There's a boatload of other proteins that have very similar structures. Not necessary that you remember all their names, but suffice it to say that these proteins have very similar structures, even though the individual proteins may play different roles in mitochondria in the IMS. So what's fascinating about D10, and we already know, is that when you have this mutation that was found in the patients, you get the aggregation of the protein within mitochondria, and this drives mitochondrial dysfunction. And so over the last few years, 3 different groups have actually made a knock-in mouse model for this, of which Veronique was one, and we rederived this in our lab. And this mutant mouse suffers from neuromuscular dysfunction at the end of its life, but it also has cardiomyopathy that happens kind of early on, and a loss of inability to continue to gain weight. So what we wanted interested in, because the heart is in some ways the easiest thing for us to visualize physiologically in terms of the function. We don't particularly care what's the heart, the liver, the brain, as Jonathan pointed out, but the heart was actually quite interesting to look at. We wanted to see by echocardiography what was going on, if it could recapitulate the phenotype that had been seen here. And so there was a reduced ejection fraction. This is also arrhythmogenic, although we don't look at this in our lab. One of the things that we could do was to look at the heart function. Now, what I'll tell you is summarize kind of what is already known about what happens when you have this mutation in D10, this S55L mutation in the mouse, is that the protein aggregates inside mitochondria, and it turns on a stress-induced protease inside mitochondria called OMA1. And OMA1 has a number of different substrates, one of which is OPA1, which is thought to be the reason why the mitochondria become more fragmented when you have this D10 mutation. But it also involves the cleavage of a protein called DELI1, which we know a little bit less about, but seems through cleavage to exit mitochondria and turn on a signaling cascade that we call the integrated stress response, which involves phosphorylation of F2 to downregulate protein translation and turn on a bunch of stress response genes like transport proteins and heat shock proteins and chaperones to try and mitigate stress condition. Now, it's as generic a title, ISR, it tells us about as much as we understand about this process, but it's something that turns on in many different types of mitochondrial dysfunction. There was reduced OXPHOS, and the cristae are dysfunctional, loss of mitochondrial DNA, and cell death sensitivity. So lots of things are going wrong. When we did the echoes in the mice, we could see a reduced LVEF in the male mice. It starts at 14 weeks of age. And so this is kind of a transition from symptomatic to pre-symptomatic. So this, when we did the echoes, or when Marcio and Armenia did the echoes, allowed us to kind of put the cursor here at 14 weeks. And so what we typically do in the lab, we like to look earlier on to see what could be the triggers, at least in the mitochondria, that are driving the phenotype. What I'm going to tell you about is only about the heart. Okay? I'm not going to talk about the other tissues. What was actually fascinating for us is when we did the same thing in the females, we didn't see anything at 14 weeks in terms of EVF or any of the other cardiac parameters we could measure. And so there was almost like a delay of this disease, as far as we could tell by echo. And this mimicked what we could see when we looked at the NPTA levels in the tissues of the mice, that it was much more strongly expressed in the males, the mutant male mice, than the females at 14 weeks, corresponding to the disease. Now, eventually, you die of what kills you first, and they all die, and roughly at the same time. So it's not that the disease is necessarily different, we think, but it's just the trajectory, at least in the heart, that's different. Now, what I'll tell you is that the protein accumulates just in the same way in both males and females, And I'll have to skip the details in terms of time, but the insolubility of the protein, which we could measure by an alkaline carbonate extraction experiment, which allows you to see the soluble proteins in the supernatant and the insoluble proteins in the pellet, happens in the males and females in the same way. So here we're just looking at the males, but you can see that even pre-symptomatically, most of D10 is soluble in the wild-type mice, but it's insoluble in the mutant mice. And so what we had here was a mitochondrial dysfunction that also correlated with a reduced, we saw at least as presented at stage 3 respiration, with reduced oxygen consumption rates that we could see here in these hearts. And it was equivalent both in males and females. So it's dysfunctional in the male and female mice at 14 weeks. Even these guys are pre-symptomatic, or these gals. And at 7 weeks, there's really no energy metabolism dysfunction, whether we fuel this with carbohydrates or fatty acids. What we could then go and do is a little bit more in the biogenetics. We could shut down the electron transfer from complex 3 and really just look at complex 4 activity, or the ability for oxygen to be consumed by respirometry. And we could see here, it mimicked what we'd seen when we just looked at the intact mitochondria and fully functional OXPHOS system, is in both males and females, you have a reduced oxygen consumption rate in these mutant hearts. So in so doing, we also looked at the cristae, and we saw this mitochondrial fragmentation and the loss of cristae. So we had all these hallmarks of mitochondrial dysfunction that can manifest in genetic models that we study in the lab. But this is also garden variety heart failure models. You'll see many of these different phenotypes, as we heard about earlier, whether it comes from a metabolic input or from an infectious input or from a surgical lesion. So we saw smaller mitochondria, which is consistent with the OMO1 activation, which we could also go on and show. And we also saw this integrated stress response turning on, or an OMO1 activation. So we wanted to look at this integrated stress response, which we could do by just looking at the levels of the proteins or that are downstream of this, or in this case, the mRNA levels. And we could see by looking at all ESR target genes that it was turned on equally in both males and females. And it was equal at 14 weeks, at 35 weeks, at 7 weeks. So the ISR was turning on equally, and the mitochondrial phenotypes were there. So there's no sex bias at the kind of molecular level, as far as we could tell. So OMO1 is a particularly interesting target because you can knock it out and the mice are totally fine. In fact, if you give mice heart attacks, or if you do genetic models of cardiomyopathy, ablation of OMO1 seems to be protective. And so we wondered whether we could actually ablate OMO1 and see what the consequence of this would be. So the way that we did this time, for a number of different reasons, is instead of knocking out the protein, we genetically inactivated with a point mutation that blocks its catalytic activity. I'm happy to go into the details of how that works. But in so doing, we crossed these mutant mice to basically knock out the activity of OMO1 in a mouse model that carries this pathogenic gain-of-function aggregating mutation in CACH-D10. And so we'd had high hopes based on previous experience to see what was going to go on. This did a really, really good job of shutting down this integrated stress response. So the hot colors here indicate that the D10 male mice have a lot of turning on of these ISR genes. And this is largely blunted if you take away OMO1. Both in male and females, and we see this by proteomics and RNA-seq as well. But it didn't fix any of the interesting mitochondrial dysfunctions that we'd hoped we would fix, right? So all these primary defects were still there, which for us suggests that maybe, well, not maybe, either the ISR is totally separate, but here it seems to be downstream of these primary mitochondrial dysfunctions, right? And again, didn't seem to be a sex bias in this response. What was particularly interesting is if we looked at a time when there was a reduced ejection fraction here in these single mutant mice, it did, even though we didn't fix any of the mitochondrial problems, it did seem to suppress or delay the onset of cardiomyopathy that we could measure by echo. Now, at 35 weeks, you get the same reduced cardiac function, whether you're a single mutant or a double mutant in red and green respectively, and eventually these guys die. They suffer the same fate and waste away in the same manner. So there's not an extension of lifespan like we would have hoped, but it was telling us something about the signaling aspect of those downstream of this mitochondrial dysfunction. But ultimately, we do believe that mitochondrial dysfunction here in this model plays an important role, because since we don't fix the mitochondrial dysfunction in the double mutant mice, and they suffer a very similar fate, we think that that's a really important feature to follow up on. So where we started was we had this D10 mutant mice with all these pleiotropic defects that start with mitochondria, but can signal outwards to the cell and to the nucleus. And in the double mutant mice, we took away this overactive innate immune, or this overactive signaling response, but we were still left with these open questions of, well, which are the mitochondrial dysfunctions that are cause and consequence? And actually, how does this work? So one of the things that we did with our single mutant mice is we actually did cardiac proteomics to try and figure out what proteins were going up and down. This is often an approach we use in omics when we don't know what we're doing, which happens often. And we did this in collaboration with Thomas Langer, who's got an excellent proteomics facility built into his lab at the Max Planck. And we know all the mitochondrial proteins. They're encoded in mitochondrial DNA in the nucleus. And so there's something called mitochondria that allows you to kind of map these. And when you look at these guys in blue, the differentially expressed proteins, the ones that are downregulated, where you see a bunch of them here on the left, which tells you that in the mutant mice at 14 weeks, here the males, there's a dramatic downregulation of mitochondrial proteins. Now, there isn't a bias about a particular compartment, and very few of them are going up. In fact, the ones that do go up are all the ones that are involved in the integrated stress response. And all these go away if you take away OMA1. And if you actually look at the pathways that are involved, there's a super long list that I don't have time to go into. But it's not so critical that you remember all these pathways. But it is interesting to know that some of the things that we picked up in proteomics also we picked up earlier in biochemistry. But here, the important thing to remember is that there's a general downregulation of mitochondrial proteins, even though our input is isolated cardiac mitochondria. So this gave us some insights that fit with the physiology we measured, but it didn't exactly tell us what was going on with this dominant negative gain of function, aggregating, and insoluble CHCHD10, which is a situation that I think about very much like this. That you've got this aggregation, and we thought, well, maybe this is a route not only CHCHD10 takes the wild type or the mutant in the mice, but many other proteins that need to be in this space. Makes me think about this. If you have a traffic jam, then you're blocking the flow of the cars that need to, or the vehicles that need to go from point A to point B. And this could have knock-on effects that are not super beneficial for the organelle or eventually the tissue. So what we wanted to do is go back to an approach, instead of doing total proteomics, actually look at the soluble proteins in the supranasal mitochondria and the insoluble proteins using a detergent-based fractionation method. And of course, looking at the total, which we'd already done. And then looking at the comparisons of these things to ask what's more or less soluble in these approaches in a totally unbiased way. And when we do this, we have a plot in which on the y-axis, you have the proteins that are insoluble because they're in the pellet, and here relative to the total input that we have. And this is important because I just told you that there's a downregulation of mitochondrial proteins when you mess around with CHCHD10. And what we look at on this left half of the graph are proteins that are aggregating more. They're more insoluble than you would expect to have in the mutant mice, in these knock-in mice. And we have proteins that we work on like MTFP1 and a bunch of other proteins for which there doesn't seem to be a particular compartment bias. So it's not like the outer membrane is more biased than the more aggregating than the inner membrane or the IMS. So there are some proteins that are on this side of the equation. I told you the CHCHD10 we know is aggregating inside mitochondria. And so we were wondering whether the proteins that follow a similar route had a different profile and were maybe potentially aggregating more or more insoluble than we expect to have. So we actually filter these proteins for ones that go in the IMS. And then here's the list that you get. So there are proteins that have these twin CX9 motifs that follow the same biogenesis route that turned out to be more insoluble in the mutant mice. And that play different roles in different pathologies or different aspects of mitochondrial biology. And there's cytochrome C that pops up here. So these two different versions of cytochrome C, which is interesting because it's what's handing off electrons to complex 4 to consume oxygen. And there's HCCS, which is involved in inserting heme into cytochrome C that it works. It can do its job in the APO enzyme. And another protein that's involved in cytochrome C. So there's a lot of data pointing to the role of cytochrome C. And so we were interested to then have a closer look at cytochrome C itself, which we do by Western. And what was fascinating for us to see is, yes, there's a dramatic loss of cytochrome C when the mice start to get sick and they have cardiomyopathy, but it's also reduced before the mice get sick, or before we can see defects by echocardiography. And this reduction that we can see here actually mirrors the, first of all, it's not rescued by OMA1, but it mirrors the turning on of the integrated stress response, which we can measure by a number of different ways, including this reporter or this ISHR protein MTHFR2. So it told us that there might be a role here for cytochrome C and we were interested to know whether this had an impact on the respiration. Since cytochrome C also has motifs similar to CHCHD10 in terms of its import pathway. So were we losing cytochrome C and was this driving the response? And so the way that we approach this is to actually take mitoplasts in which you rip off the outer mitochondrial membrane and measure the respiration or measure the oxygen consumption rates. Here, adding TMPD ascorbate to basically cycle the redox of cytochrome C. And we could see here that the mutant mitoplasts had a reduced respiration rate. And if you add in cytochrome C from the cow in this test tube, you can actually fully rescue the oxygen consumption rates, which for us told us that there's an underlying cytochrome C deficiency that's involved in CHCHD10. So our current model, and it's still en route, it's still a work in progress, is that D10 seems to aggregate and might impinge upon this import pathway that's important for the import of many different proteins, including cytochrome C. And this biogenesis is one of the reasons why things are going off the tracks. We have no idea whether this is true for the other tissues that seem to be affected in this mouse model, but it's something that we're following up on. So with that, I'd like to thank the people that did the work, notably Marcio and Armenia, who's no longer in the lab, who've been driving this project with fantastic technical assistance from Elodie, the funding sources, wonderful core facilities here in-house, and our collaborators in Nice, who made the landmark discovery of mutations in CHCHD10. Thank you.
Tim, thank you very much. That was a fantastic talk and really interesting. So as we wait for questions in the Q&A chat box, I guess I'll go ahead and start. And I'm just wondering, the cytochrome C is really interesting. Do you see that it stays in the intermembrane space and it's actually not going to the cytosol to trigger apoptosis? Is that something you've looked at yet? Because
we haven't looked at
that would be
this.
like insoluble cytochrome C that's not moving from the intermembrane space.
Well, there'd be less of it, and it wouldn't be necessarily leaving. So we haven't looked at this. We don't see any real evidence of we don't see cardiac troponin go up at any point in the mouse, right? So we don't know about apoptosis in the cardiomyocytes in this model. But it's not a phenotype that we see. So we're a little bit surprised. And what is interesting is in the cells from the patients that have these mutations, the fibroblasts are reported to have less apoptotic sensitivity. And that was never explained. It didn't make any sense. So I mean, it's interesting to hypothesize that perhaps this reduction in cytochrome C blunts respiration, but it could also have an impact on reducing apoptosis, and whether that promotes then a shift to more inflammation, we don't know. But we haven't explored that much further in the heart.
Okay.
Can I ask a question too?
Yeah, go for it. Go for it, Christoph.
Yes. So with the integrated stress response, we also observed this in our Barth syndrome model with tafazin deficiency. And although we do not know for sure the causality, whether it contributes to a maladaptive or adaptive metabolic rewiring, we do see the rewiring of metabolism. So, and we assume that this is a protective response in the first place that's trying to compensate for some of the mitochondrial deficiencies by providing extra mitochondrial sources or alternative mitochondrial and extra mitochondrial sources of ATP, but also glutathione, for instance, and antioxidation. So do you have any hints whether in terms of metabolism, so substrate metabolism, you have shifts or alterations? For instance, we observe that amino acid metabolism is compensatorily increased in Barth syndrome, and glutamate respiration can even go up with this. So do you see anything indicating that this is affected in your model as well?
So when we look at the double mutant mice in which we've blunted the ISR, we have no proteomic differences other than the ISR. So there's no change. We have no evidence there's a difference in the metabolism between the single mutant mice and the double mutant mice.
Mm-hmm.
So we don't always get so OMA1 has other substrates, right? But if you take away the ISR using any sort of means, even the mitochondrial arm of it, it's not entirely clear depending upon the pathological situation whether it's good or bad. And I think it really depends. And we have situations in which you can take away OMA1, you have still the ISR turned on, not in the heart, but in other tissues. What I think is interesting, we're thinking a lot about glutathione because this process is obviously GSH dependent, right? And so what I don't fully understand in our particular model, and this is based on glutathione, is why is it only certain tissues that are affected? And so the protein doesn't accumulate, it doesn't aggregate in the liver, it doesn't do it in the kidney. Like there's no, it doesn't make any sense other than perhaps you need a certain redox stress or a glutathione insufficiency, let's say, either in the IMS, which comes obviously from the cytosol through GSR and GS glutathione reductase and other inputs. So we're thinking a lot about glutathione levels and how that might be different in different tissues, but nobody's ever looked at that in vivo with probes. They've only done this in yeast. So no is the answer to the differences in metabolism as far as we can tell, but we are paying specific attention to glutathione metabolism, which does come up to be something that seems to be sex dependent also in the heart.
Interesting, yeah. Thank you.
Okay, we have a question from the Q&A box. It's from Thomas Pulinil-Kunil. What is the effects on CHC mutation and or deficiency on lipid utilization? A prior study showed that high-fat diet feeding attenuated cardiomyopathy in CHCHD10 mutant mice. Do you think forcing FA utilization might offer benefits?
Yeah, so it's interesting because these mice don't gain weight as much. And there was a beautiful study from Giovanni Manfredi's lab in which they gave the mice a high-fat diet and miraculously they did better. Like they rescued the cardiac function. And it also rescued in this model the protein insolubility. So those blew our mind. So the short answer is we've never looked at this. We did do an experiment where we tried to, we put them at thermoneutrality. And the idea was maybe we could lower the heart rate and maybe things would get better. And the answer doesn't fix the problem. But the mice did get fatter. The wild type mice, which of course makes sense, but the mutants did. So what I do think is there's probably organ crosstalk that we haven't fully understood. And this comes back to what Lisa is working on in her team. And it would be interesting to look at the organ crosstalk in vivo to understand the lipid utilization. So I think that the beneficial effects of a high-fat diet in certain, which we've also worked on as well, in certain cardiovascular contexts is still controversial and not fully understood. We still need to do more work in understanding the lipid metabolism in our system. Now, what I can say is palmitoylcarnitine oxidation rates are identical in between or reduced in the mutant mice and aren't fixed in the double mutant mice. So we think that there's a general mitochondrial problem, and it's not necessarily beta oxidation problem per se that's been caused by this cytochrome C deficiency.
There's one more question. So from Lobby. So do you think there could be a differential accumulation of the protein in dividing cells versus non-dividing cells, like neurons and cardiac muscles?
Possibly. You can get this protein to this particular version of the mutant protein to accumulate in postmitotic cells as well as mitotic cells. I personally think that the rate of accumulation of this protein is probably going to be a function of the redox status in the IMS and in the cell. So, and then the question becomes, is the redox status in different cells different? Probably the answer is yes. So I don't know that it's so simple to say it's dividing cells and not dividing cells. High levels of this protein have been proposed to actually reduce cell proliferation. So it could be a chicken or the egg situation. But I don't know that it's. I don't have a clear way of how I could explain it's the dividing or postmitotic versus mitotic cells. I think it's more the redox that's going to be important.
And then just when these aggregates form, do you see like more mitochondrial derived vesicles? Is there a way the mitochondria are actually trying to clear these aggregates out? Is there more peripheral division than midline division of the mitochondria? Have you looked at any of this?
Yeah, so we've looked. I can say in our hands, we don't see that the mutations, patient-derived in patient cells, we don't see any effect on mitochondrial morphology. So I think that the mitochondrial fragmentation and the changes in crystal that we're seeing in our model is very, very difficult to know whether that's because the heart is doing poorly or because it's actually the D10 that's aggregating, like, so, cause or consequence. And so we're not at that stage yet. How it's dealing with these aggregates, I'm not sure. I think that perhaps what's happening is the cells are dying, but we have never looked to see whether these aggregates are leaving. Now, what has been reported in some tissues is that these things can be found outside in the myocardium and other tissues. So does that mean it's that the cells exploded and now you find these aggregates? Nobody's done these kinetic analyses in vivo. So it's very difficult to know.
Okay, well, I don't see any more questions. So Tim, thank you so much for the fantastic talk. Also, Lisa and Professor Mack. So everybody, don't forget that March 15th is the deadline to submit for the Young Investigator Travel Awards and for your abstracts, so everybody can go to Japan. Hopefully we see you there. So I think we'll sign off. Caroline, is there anything else? Nope. Okay, so I think we're done. Thank you very much, guys.
Thank you, guys.
Bye.
See you, Nara. Bye-bye.
Thank you. Bye.